Great. So thanks for coming. I will tell you about some new methods we've developed for nucleic acid analysis, as the title says, using tensor networks and random matrices and some other ideas from dynamic programming and discrete math. So I think it should be apparent to everybody that RNA and DNA are pretty important to society at large, but also the research here at the lab. So just some of the example applications that nucleic acid design analysis has been used for are pictured here, and these are some from my collaborators. So first, detection of SARS-CoV-2 or other viruses can be done itself using RNA and DNA that's engineered in a specific way. Some of the applications that I've been involved in are imaging gene expression within developing organisms. So here there's a couple plots of different mRNA expressions in a multiplexed experiment within a chick embryo or a mouse brain tissue. But we can also get farther afield and do many different things with RNA and DNA. So within the analog computing paradigm, you can actually use DNA to perform biological computing. So you can perform matrix multiplications or square roots or neural networks even using DNA in sort of a chemical reaction network paradigm. But when we think about modeling, analyzing, or defining DNA and RNA, we can think about it in terms of three basic levels of structure. And this is roughly the same way people think about proteins, but it's relatively unique in how it works for RNA and DNA. So first of all, we have the sequences that I think we're all familiar with. So these are basically RNA and DNA are strands of fixed sequences of nucleotides. So ACGU for RNA and ACGT for DNA. These are fixed on the timescale of an experiment. What's not fixed is the secondary structure that RNA and DNA can adopt, or its tertiary structure. So the secondary structure, there are a whole host of ways to plot it, but it basically refers to this discrete set of hydrogen bonding base pairs with that complex. So if you have a sequence, those are covalently bound nucleotides, and you can have base pairs between any bases in that sequence. And those can fluctuate on the order of microseconds or less with every experiment. And then at an even more detailed level, there's all of the tertiary structure details of a biomolecule. So all of the atomistic configuration and so on, more detailed degrees of freedom. What's unique about RNA and DNA in particular is that they're extremely programmable because they can be understood very well in terms of secondary structure alone. So compared to proteins, there's been a lot of successful work using discrete math to attack secondary structure analysis in particular, and that's what I've worked on. So even though secondary structures is a highly simplified way to view nucleic acids, it can still be pretty difficult in a computational method. So the reason is that a secondary structure can be thought of as a matching on the graph of all nucleotides within a sequence. So if you consider all possible sets of matchings for a sequence of significant size, that quickly becomes combinatorially large. So the number of structures that something of 100 nucleotides can adopt is like 10 to the 24. The number of structures that SARS-CoV-2, so something like 30,000 nucleotides can adopt, is like 10 to the 7,000. So it quickly becomes completely infeasible to even think about enumerating the ensembles of such sequences. However, by restricting structures that we're interested in to just connected and non-crossing structures, which is a particular configuration that I've illustrated on the top and bottom in an equivalent way, we can actually sum over all structures within this exponentially large ensemble using exactly using order n cubed programming. However, when we look at dynamic programs in the existing literature, which I have worked on, the free energy models at the bottom of these dynamic programs are highly complicated and rather suspect in many significant ways. So they've been developed over decades of experimental and theoretical work, including many heuristics and special cases and how the free energies are actually formulated. And what this results in algorithms is a huge mess of different recursions that have to be hand-coded and developed with pretty complicated interactions with them. And this has led to a state that's pretty rigid and slow to extend to new materials and new data. In contrast, we've posed a different model based solely on tensor networks. So in this, it's a very natural mapping from a secondary structure, like is plotted on the left, equating each of the loops, which is like a face in this graph, with a matrix product trace to compute its partition function. And then just by some formalism of pre-contracting matrices, we can get to a diagram like on the right. So here, each of the nodes is actually a tensor, and then every edge between the nodes is an index that's contracted over. And the specific way that on-crossing partitions show up in these tensor network diagrams means that they can be contracted over in layer time. So in further work, we've demonstrated not only that we can replace dynamic programming algorithms in a very elegant way with such formulations, but we've also sought to replace dynamic programs entirely. So dynamic programs are a paradigm that's a little complicated, but essentially for this works in order and cube time, so it scales cubically with the length of the sequence. What we've demonstrated is that there's a pretty deep and interesting connection with fields of random matrix theory and free probability. So actually, if one works out, if one computes a trace of a product of random matrices in a particular way, we can actually embed our exact model, including all of the complementarity and free energy relations, into this randomized estimator, which appears as a simple matrix product. So this gives us an asymptotically exact summation inference for the full tensor model, just using the simple formulation and no dynamic programming whatsoever. So putting this together, we've tested this and benchmarked these models on a whole host of problems, ensured they're more powerful than current models. So from old models on the left, we can fit experimental data with better accuracy with many fewer parameters and with pretty small sizes for the tensors involved. And then because everything has been boiled down into such a computationally simple paradigm, using these matrices and tensor algorithms, we can enable GPU computation for the first time for these sorts of algorithms. And so this, for large sequences, allows up to roughly 200-fold speedups over a serial CPU approach. So with that, I hope I've demonstrated how some of these surprising and new methods are impacting nucleic acid analysis. So I brought in tensor networks and random matrices in a fundamental way to affect these problems. And I think this has led to some new paradigms that have future applications. So one-shot design, randomization, and extension to different materials. And then on the performance side, our models have also proved to be fast and accurate. And then we've also programmed them on a GPU for the first time. So that'll thank my collaborators. I was Pierce at Caltech, and Michael Lindsay here at Berkeley.